In lipid digestion, lipids, when we take as a mixture of different lipids. Okay, it may be triacylglycerol, it may be phospholipid, it may be cholesterol or fatty acid, or monoacylglycerol, diacylglycerol, again cholesterol esters. So these may be the component when you take lipid, but 90% of the ingested lipids are triacylglycerols. Major component is triacylglycerol. In metabolism of lipid, we will remember some key words. Emulsification, bile salts, Pancreatic Lipase, mixed micelles, Re-esterification, Chylomicrons. Can you see from backside? Okay. Now, lipid we take together in large say droplets. Okay. Now, these lipids are not suitable for enzyme activity. For better digestion, these has to be broken into small droplets. So what they do, what is done? It is broken small lipid droplets. This is done by the bile salts. Indian diet, on an average, it contains 20 to 30 gram of lipid per day. But nowadays, it is not so less. Even it may go up to 150 gram per day. Those who are taking fast food, they are consuming a lot of lipids. Okay. Now, when we take lipid in the mouth, each stay in the mouth is very less. Again, in the mouth, from the dorsal surface of the tongue towards the back side, the enzyme for lipid digestion is called lingual lipase is secreted. Okay. Now, as it is, its stay in the mouth is very less, and it is secreted in the dorsal surface of the tongue. As soon as it is secreted, almost immediately it goes to the stomach. Second point is optimal pH of activity, lingual lipase activity is 2.5 to 5. Now, stomach having acidic pH, and that is suitable for the lingual lipase activity, and it is also called a acid stable lipase. So, enzyme therefore continues to be active in stomach. It usually acts on short chain fatty acids present in milk, butter and ghee. Now, as soon as the food particle containing lipid enters in the stomach, the gastrin hormone is released from the stomach. Okay, and that gastrin stimulates release of gastric lipase. Now, wonderful thing is gastric lipase, it is secreted in the stomach but has no activity in the stomach. It has to work below down in the intestine. So, major digestion of lipid occurs in the intestine. Okay. It is secreted by the chief cells, stimulated by the gastrin. 30% of digested triglyceride occurs in the stomach, but major is secreted or digested in the intestine. Now, here comes the role of emulsification. Emulsification is provided by the bile salts through its detergent action. Emulsification is provided by the peristaltic movement of the mechanical mixing. In a bottle, you add oil and some amount of water, shake it. You will see that it becomes milky. That means lipid particles, they become very tiny droplets, and that will be emulsified due to mechanical movement. So, peristalsis, it causes mechanical mixing of the bile salts and lipid along with the aqueous medium, in the aqueous medium giving better emulsification. Then phospholipids, these are the amphibitic lipids. So, they also, what they do, they bind on the lipid surface and split it. Okay, bind on the lipid droplets surface and they split it and make smaller droplets. Bile acids, sodium glycocolate, sodium taurocolate, they reduces surface tension, emulsify fat droplets, increase the surface area of the particle, enhance activity of the enzyme. So, more the droplets are small, larger the surface area. Okay, this was you understood, no? Smaller particles, when same amount of lipid molecules, they are, can you see this from this side? When they are broken into a numerous number of small droplets, the portion that contains smaller droplets will have larger area, because large droplet you have only this much surface area. Smaller droplets you have this one also, this one also, this one, this one, this one, this one. So surface area increases, no? That's why this surface area will have enzyme action. Hydrophobic portion of the bile salts intercalates into the large aggregate of lipid with the hydrophilic domain remaining at the surface. This leads to breakdown of large aggregates into the smaller droplets. The surface area of the action of lipases increases. See, this is a large droplet. These bile salts they bind like that and split this one and make a smaller droplet like that. A multiple smaller droplets are produced, small droplets are produced, making emulsion. Now, from the stomach containing partially digested triglyceride and other undigested lipid particles along with the other food passes to the intestine. This semi-liquid diet is called chyme. When this chyme passes to the intestine, in the amino acid metabolism, we read, not metabolism, amino acid digestion of protein, we read two hormones are secreted, CCK and secretin, cholecystokinin and secretin. So what happens? So, food enters here, it releases CCK and secretin. CCK2 does two things. One is it stimulates pancreas to secrete pancreatic juice. And it stimulates gallbladder to secrete bile. Bile contains bile acids and bile salts. Our interest is with the bile salts. Okay? So, bile salt, it has a role of making the ingested fat into a good emulsion. Okay? Pancreatic juice, it contains pancreatic lipase, phosphodiesterase, phospholipase A2, cholesterol esterase. These are the hormones of lipid digestion. They are secreted from the pancreas. Another thing is secretin. It causes pancreas to release bicarbonate. So the intestinal chyme, which is acidic, it is neutralized to neutral pH or towards the alkaline pH. Now, which is the required pH for better function of pancreatic lipase and other lipases. Okay. Now, this pancreatic lipase, it acts on triacylglycerol containing long chain fatty acid. and the gastric lipase, lingual lipase, they act on the short chain fatty acid and medium chain fatty acid. The lipid containing short chain fatty acid and medium chain fatty acid. So, milk, mother's milk, human being contains short chain fatty acid. And infant in the stomach, they are not having that strong acidic pH. This is almost neutral pH in the stomach of infant. So, in the stomach, the action of lipases, that is lingual lipase and gastric lipase, they completely hydrolyze ingested milk fat. So, it is beneficial for the infant for digestion of the mother's milk. Now, in case of lipids containing long chain fatty acids, they are acted upon by the pancreatic lipases. Phospholipids, they are acted upon by the phospholipase A2. And cholesterol, they are acted upon by the phosphocholesterol esterase. So, now, in case of pancreatic lipase, it is a peculiar character. In case of triacylglycerol, so in carbon 1 there is one fatty acid chain, carbon 2 there is one fatty acid chain, and carbon 3 there is fatty acid chain. So pancreatic lipase, it acts on first on the number 1 carbon and releases, this is the triacylglycerol. On first carbon they act, it will release 2,3-diacylglycerol and free fatty acid. Okay? Then again pancreatic lipase. Then again pancreatic lipase will act, release free fatty acid and again it will act on the third carbon, releasing the free fatty acid and 2 monoacylglycerol. Thus one molecule will give 2 free fatty acid and 2 monoacylglycerol. After that, what happens? This free fatty acid, this isomerized to shift in the position 1, carbon position 1. So it becomes 1 monoacylglycerol and again acted upon by the pancreatic lipase to release completely glycerol and 3 molecule of fatty acid. Now here it is the triacylglycerol. It will act on release 1 fatty acid from the group 1, 2,3-diacylglycerol, then 2 monoacylglycerol and fatty acids. Okay. Now after digestion, we will get what? We will get 2 monoacylglycerol, 1 monoacylglycerol, glycerol and fatty acid. This is the product of digestion of triacylglycerol. Okay. Now all the triacylglycerol is not converted into glycerol or fatty acid. So that's why it is incomplete, because some amount of monoacylglycerol will be there, some amount of diacylglycerol is there, and glycerol, fatty acids are there. Now this glycerol, okay, I'll later on I'll tell. Now cholesterol ester, it is acted upon by the cholesterol esterase enzyme. Cholesterol esterase enzyme releasing free cholesterol and fatty acids. Phospholipase A2, it releases phospholipid, lysophospholipid and fatty acid. From the second position of the phospholipid, the fatty acid is removed, so the lysophospholipid is formed. Here 1, 2, 3, in first and second carbon there are fatty acid, third carbon phosphoric acid plus nitrogenous base. So acted upon by the phospholipase, from the second position the fatty acid is removed. Now the rest is lysophospholipid. Here lecithin is one of particular name of the phospholipid, so it is lysolecithin. It is the nothing but the lysophospholipid. Okay. Now so it is again added there in the intestinal fluid for further action. Action means for absorption. Cholesterol acted upon a cholesterol esterase, and the free fatty acid from the hydroxyl group is released, and there will be free fatty acid and free cholesterol. Now one thing is that the bile contains bile salts plus bile acids. These bile acids, they have a inhibitory action of pancreatic lipase. They do not allow pancreatic lipase to act upon the lipid molecule. So what happens? Another protein of about 12,000 molecular weight is released from the pancreatic juice, along with the pancreatic lipase. Now function of this is they bind to the triglycerol molecules at the oil and water interface, obligatory for the action of lipase. So they say this is the triacylglycerol and this is the enzyme. So they bind here. And do not allow the bile acid to come into play in the reaction. Inhibitory role in the reaction. So that's why their action is very much required, otherwise digestion is not complete. This co-lipase is secreted as zymogen, pro-co-lipase. When it is activated, how it is activated? It is activated by the trypsin. Like other proteolytic enzymes which are secreted as zymogen, acted upon by the trypsin and made it active after removing few amino acid from their polypeptide chain. So likewise, in same way, they are activated by trypsin. monoacylglycerol, lysophospholipid, cholesterol, free fatty acid. Now they are ready for absorption. Long chain fatty cholesterol, phospholipids, lysophospholipids are incorporated into molecular aggregates to form mixed micelles. Mixed micelles, it is again a very important molecule for absorption of digested lipid. Mixed micelles are the spherical particle with a hydrophilic exterior and hydrophobic interior. Now, these cholesterol free fatty acid, these are the strongly hydrophobic. They lysophospholipid, not cholesterol. They come in the center of the molecule. Then bile acids, bile salts. Bile salts, they will be in between so bile salts, due to their detergent action, help to form micellar aggregates. Now, phospholipids. They will be arranged in this way. In between, there will be cholesterol molecules. Now, these micelles, they come into contact of jejunal epithelium of the jejunal epithelial cells. Jejunal epithelial cells, then fatty acids, monoacylglycerols, and other digested product passively diffuses into the mucosal cells by simple diffusion. Simple diffusion, they enter in the cell, microvilli cell. They are in the microvillus, inside the cell. The re-esterification of molecule occurs, such as from monoacylglycerol, two molecules of free fatty acids are attached, forming triacylglycerol. From phospholipid, fatty acid is attached to form, from lysophospholipid, phospholipids are formed. Okay? Cholesterol are esterified into cholesterol esters. Then free fatty acid, it remains as some of the free fatty acid. So they are again re-esterified to the original product, triacylglycerol, phospholipid, cholesterol esters. Now the glycerol which was digested in the process of lipid metabolism from intestine, it rapidly absorbed by the intestinal cell and released in the bloodstream. So the glycerol molecule which was digested is not available for re-esterification. Okay, so that very glycerol is not present for re-esterification. So this glycerol which is synthesized inside the cell is totally different. Okay, so now we have triacylglycerol, some of the monoacylglycerols, some long chain fatty acids, some diacylglycerols, then cholesterols. They are inside the cell ready for absorption. Now how it is absorbed? It is absorbed by formation of chylomicrons. Inside the intestinal mucosa, long chain fatty acids are re-esterified to form triacylglycerol. Fatty acids are first activated. Fatty acids are first activated to fatty acyl CoA by acyl CoA synthase enzyme. So a coenzyme fatty acid, before taking part in any action in metabolism also, it has to be activated to fatty acyl CoA. Okay, now fatty acyl CoA is the active form of the fatty acid. Fatty acyl CoA by the enzyme fatty acyl CoA synthase, then acyl CoA synthase needs to lysis of the two high energy bond. Now formation of this needs utilization of 2 ATP for each, adding up for each fatty acid with the monoacylglycerol. This is used, ATP is used. So now it is clear that monoacylglycerol is the precursor for synthesis of triacylglycerol. But cell can supply glycerol synthesizing from the glucose, but not the glycerol which is digested in the intestine. That will not be used up in here. Free glycerol absorbed from the intestinal lumen is directly. So free glycerol is not available for re-esterification, but the cell can convert glucose to glycerol phosphate and synthesize triacylglycerol. Now just schematic diagram. In jejunal epithelial cell, in the lumen mixed micelle is formed, bile acid is released, other things like triacylglycerol, fatty acid, 2 monoacylglycerol, 2 means position 2. Okay, monoacylglycerol, short and medium chain fatty acids, all are absorbed here. And bile acid is released in the lumen. It is absorbed from the terminal part of ileum, and it is reabsorbed in the circulated to the liver again. There it stimulates again bile acid secretion or bile secretion. This is called Chylomicron action of the bile salts. So now these things inside the cell inside the cell, they are ready for absorption, and absorption is facilitated by forming chylomicrons. Chylomicrons, they are absorbed. How? Chylomicrons, they are brought to the periphery of the cell, and through exocytosis, the material is released in the lacteals. These are the lymphatic system. This is the lymphatic system. These are carried to the thoracic duct, and from thoracic duct to the vein. Then they are carried to the enterohepatic, to the liver and peripheral tissue. Now, what are chylomicrons? Short note. These are the incorporated into chylomicrons are triglyceride. Triglyceride is there, cholesterol esters, phospholipid, apoprotein B48, apoprotein A. The chyle, milky fluid from the intestinal mucosa cells loaded with chylomicrons are transported through the lacteals into the thoracic duct and then emptied into the lymph circulation. This is a picture of schematic diagram of chylomicron. The core of this chylomicron is constituted by triacylglycerol and cholesterol ester, not phospholipid. I told you phospholipid. It's not phospholipid, cholesterol esters. Cholesterol is, you have two molecules are there. One is cholesterol, another is cholesterol ester. Cholesterol ester is strongly hydrophobic, so it comes in the center. Triacylglycerol, it comes in the center. In the periphery, these are the phospholipids. These are the phospholipids, these are the cholesterols. Now you can see, you have seen phospholipid in the cell membrane also, constituting the cell lining. Okay, now cell membrane, making the cell membrane. Cholesterol also is there in the cell membrane. Now what is the difference between chylomicron and cell membrane? In cell membrane, the phospholipids are bilayered. Here monolayer, but cholesterol also there, like cell membrane. So difference between the cell membrane and chylomicron is here phospholipids will be monolayer. Now some proteins will be there, ApoB48, important constituents of the chylomicron, ApoB48, ApoC and ApoA. So now this chylomicron, when serum may appear milky after a high fat meal due to the presence of chylomicrons. Chylomicrons, they will be released into the lacteals by exostosis. It passes to the through the lacteals, then to the thoracic duct, then to the inferior vena cava, then it comes into the blood. So from the blood, the respective tissue will take it up. When there is a high content of lipid, amount of chylomicron they increase, and serum becomes milky white. But usually it is cleared up within few hours. This is a chylomicron assembly, diagrammatic representation of chylomicron assembly. Fate of chylomicrons, the absorbed triacylglycerides are transported in the blood as chylomicron. They are taken up by the adipose tissue and liver. Liver synthesizes endogenous triglycerides. They are transported as VLDL and are deposited in the adipose tissue. During the starvation state, triacylglycerol in adipose tissue are hydrolyzed to produce free fatty acid. Free fatty acids is activated, then it takes part in the beta oxidation to give energy. Short chain fatty acid, they do not require anything. They are, after digestion, they are absorbed, they are taken up by the intestinal cells, then they are released in the blood. Short chain fatty acids are predominant constituents of milk, butter, ghee. Medium chain fatty acids are coconut oil and mother's milk. Do not need. See, milk, cow's milk and mother's milk, they contain both short chain fatty acid and long chain fatty acid. Short chain fatty acids in mother's human milk, it is more, and in case of cow's milk, it is medium chain fatty acid. But different books write different thing. Do not need re-esterification. These fatty acids do not need re-esterification. They directly enter into the blood vessel, then portal vein, finally liver, where they are immediately utilized for energy. their absorption is rapid. No extra thing is required. Directly they can get absorbed. They are better absorbed than long chain fatty acid. And here is also just a picture of how the chylomicrons are formed and transported in the dictyals. Abnormalities. So defective digestion, steatorrhea. Steatorrhea means passing of lipid in the stool when there is no bile salts in liver disease. So emulsification is not complete. So there is defective digestion. Excessive fat is passed in the stool. Then pancreatic disease. There will be inadequate synthesis of pancreatic lipases and other lipases. So there will be steatorrhea. Steatorrhea, and in pancreatic disease, chronic pancreatitis, sometimes the condition gives a better, it becomes a bit better, then again it goes worse, like that. Sometimes there will be passing of steatorrhea, then sometimes it is okay. This alternate okay and steatorrhea is called split steatorrhea. Okay. Gallbladder obstruction, stone in common bile duct doesn't allow bile acid to pass in the intestine, so there is no emulsification. There is problem in lipid digestion and steatorrhea will be there. Celiac disease, this is sensitivity to gluten, so that will cause immunological destruction of the intestinal cell, and that will cause steatorrhea. Gallstones, tumor head of pancreas obstructing the pancreatic duct, so pancreatic enzyme cannot come, bile cannot come. Enlarged lymph node giving the pressure, not allowing the fluid to come. So chyluria, these are not actually digestion problem. Chyluria means when there is an abnormal connection between the thoracic duct and urinary bladder. So chyle passes with urine, milky urine, and chylothorax, when abnormally thoracic duct is opened in the pleural space, there is collection of this one chyle in the pleural cavity. So that is chylothorax. So with this we will complete digestion of lipid.